Hey everyone, welcome back to AS and A-Level Biology with Dr. Demi. In this video, I am continuing with the exam prep by doing the May June 2021 exams. And as promised, this is paper 4. For those of you who might be watching this video for the first time, I am simply revising the exam the May June 2021 cohort had to write. And I'm only going to be doing papers 1, 2, 4, and 5, variant 1 of each paper. For those who keep asking about paper 3, please know that paper 3 is a laboratory-based paper, and so for that reason it is difficult to simply just record it on a slide and put it on YouTube. You need to do the practicals, get the data, and then walk through it. So I would suggest that you ask your teachers to help you with that. For paper 4, which is usually the first paper, if you're writing the May June cohort, that is, it's usually the first paper. I'm not sure if it's the same with the October-November cohort, but it's a paper for 100 marks. So you have 100 marks for paper 4, and you have 2 hours to write it. So it's a very long paper, and it contains, it always takes content from chapter 12 all the way to chapter 19. All of these chapters are important because I notice students also usually ask, well, which one should I study and which one should I not? Please do not skip any of these chapters at all. You need to study every single one of them and be sure that you understand, because it's mostly about application of knowledge as opposed to you just being able to memorize stuff and regurgitate them. Of course, you do get some of those questions, but that's not the default mode for this paper. Because this paper is so long, I don't think I will record everything in one video. I'll see how it goes. I might record half of the paper first and then do another half in a separate video, but that will depend on how long the video starts to get as we go along. So let's get into it. This is paper 4, May June 2021, variant 1, and these are the solutions. So first question, photosynthesis is a complex process involving the transfer of light energy into chemical energy. Describe the role of photosynthetic pigments. So again, if you've watched the videos, you already know what this is about. And obviously if you've been going to class as well and listening in class. And also just to say before I even answer this question is that please note that this channel is not a substitute for your classroom teaching. I am not trying to take the place of your teachers. I am simply providing you with a tool to revise what you've already learned in a more accessible format to you that might be easier for you to just come back to whenever you want to. That is the purpose of this. So there's some things that might be covered in your classroom that will not be covered here. The responsibility is on you to make sure that you follow through and check that all your checks and balances are in place for your revision and for your examination preparation. So what are the roles of photosynthetic pigments? If you remember from photosynthesis, we have what we call the primary pigment, which is either chlorophyll A or chlorophyll B, chlorophyll A or B. And then you have accessory pigments, some of which are like your carotenoids and so on. So the point of these pigments is, first of all, is to trap light, right? So they harvest light from the sun. And if you watch the video I did on photosynthesis, you'll be able to get this. They harvest light and then they ensure that that light goes to the primary pigment, either through cyclic or non-cyclic phosphorylation. So in this case, again, I always tell students, please don't write too many unnecessary stories. You don't need to write full sentences as well. This is a biology paper, not an English paper. So you can just say they harvest light from the sun. They enable the cyclic phosphorylation or non-cyclic phosphorylation process to take place. They pass energy. So the accessory pigments pass energy from the sun down to the primary pigment. You're only required to write 2 things. That's why it's 2 marks. So any of those 2 would suffice. Name the precise location in the chloroplast of photosynthetic pigments. So you always find photosynthetic pigments in the thylakoids, or you can also say you find it in the grana. Again, if you're not aware of this, please watch the video I did on photosynthesis. By the way, I'm not going to write all the answers out. I'm just using that to, while I explain, if I'm able to write it out, I will. But if I can't, please just listen and take note. Name a practical technique that's used to separate photosynthetic pigments. That technique is called chromatography. If you haven't done this experiment in your classroom, then please ask your teacher to set it up. It's quite easy. But what my students would typically do is that they would take a bit of spinach and they would grind it in a solvent, and when they grind it in the solvent, they then put it in they put it on chromatography paper, and then they put that paper in a solvent, another solvent, and what you will see is how the solvent moves up and separates the pigments into different colors. So you'll be able to see that. So please ask for this to be set up, or if you don't know how to set it up, or you're not able to set it up at your school, just watch videos on chromatography online. Let's look at B. The rate of photosynthesis is affected by many environmental factors. A student carried out an experiment to investigate the relationship between the concentration of carbon dioxide available to an aquatic plant, Cambomba caroliniana, and its rate of photosynthesis. And this is what the setup looks like. And then they go on to tell us sodium hydrogen carbonate solution was used as a source of carbon dioxide. The concentration of CO2 was varied using 6 different concentrations of sodium hydrogen carbonate solution. All the plants were kept in the dark before the lights were switched on at the start of the experiment. 5 replicates were carried out at each concentration. The rate of photosynthesis was obtained by calculating the percentage change in dissolved oxygen concentration in the solution over 5 minutes. So again, you can just come here and look. I know I already pushed the animation button, but you can see over here, this is, I believe, the plant, and then the plant is put in that solution, and that solution is supposed to provide the plant with CO2, and it's a sealed container, and obviously you have light. So this is more or less a photosynthetic experiment. One thing I want you to bear in mind before I change the slide is that photosynthesis can be described by this reaction over here. It is CO2 plus water to give glucose, okay? Because you make plants make sugars when they photosynthesize, plus oxygen, which is given off as a gas, and this obviously has to be 6 CO2. All right, so just bear that in mind. What this is basically saying is that they have fed this plant sodium hydrogen carbonate solution as a source of CO2. So the plant is getting CO2, and obviously it's in solution, so it's also getting water, and they're now measuring the rate of photosynthesis by the change in oxygen. So they're more or less measuring what the plant is doing. All right, and these are the answers or the results that they got. I hope that it's clear enough. You can see it. So obviously at over here, concentration of sodium hydrogen carbonate solution at 0 concentration of that, your rate of photosynthesis is more or less 0. There's a point here that indicates a negative change in the oxygen, which we will get to. And then as you can see that as the CO2 concentration increases, so does the rate of photosynthesis until it gets to a certain point where things flatten out. So let's see what the first question says in this case. It says, with reference to this figure, explain the pattern of results obtained between 0.25 and 1.25. So explain what has happened here. Now again, as you can see, the rate of photosynthesis steadily increases until it gets to a certain point where it flattens off. So what you can do in this case is you can talk about how you can talk about limiting factors actually in this case. So you can say, well, the more you supply CO2 to the experiment, CO2 becomes available in abundance, right? So it's more like the enzyme question as well, where we spoke about how the rate of reaction of an enzyme would flatten off even if you keep increasing substrate concentration after a certain point. The same thing happens with photosynthesis, except you can't use the exact same explanation. In this case, we now have an increase in CO2, and obviously as we increase it in bits, the rate of reaction is increasing. But it gets to a certain point where the CO2 is available in abundance, but other things start to become limiting factors. Because, for example, for you to have your photosynthesis taking place, your CO2 might need to react with water. You need light intensity for that. Your CO2 might be binding with what's called ribulose biphosphate. Temperature might be affected. So basically all you need to explain here with reference to this image is that between 0.25 and 1.25, the rate of reaction increases. The rate of photosynthesis increases steadily until it flattens off. Then you will need to explain that the reason why it has flattened off is because there is an abundance of CO2, which means CO2 is not a limiting factor anymore. And then you can mention one of the other factors that affect the rate of photosynthesis as a limiting factor. So that could be light intensity, temperature, or any other thing. Let's look at the next one. The percentage change in dissolved oxygen for that plant at 0.0 is negative. So what would be the reason why it has a negative value? So obviously, if it has a negative value, it means that the plant is not undergoing photosynthesis, rather the plant is undergoing respiration. So remember that with respiration, we flip the equation for photosynthesis. This is the formula for respiration. I know everyone likes to start with glucose, so I've sort of started off differently here, and then you get CO2 and water. So this is respiration here. Okay, 6H2O. This is the formula for respiration. So if there is no photosynthesis happening, because as you can see, the concentration of CO2 is 0, and percentage change then comes down here to about -10, what that tells you is that the plant is using oxygen. So if the plant is using oxygen, then it means the plant is respiring, and so that is what you'll be able to say there: no photosynthesis and respiration using oxygen. All right, let's look at this one. To minimize temperature changes, the student decided to use an LED lamp as a light source. LED lamps release very little heat energy. Explain the importance of minimizing temperature changes in this experiment. So in this case, what you're dealing with, like it says, when you use an LED lamp, you release very little heat energy, which means that there would be no temperature change or negligible temperature change with your experiment. So the importance of minimizing temperature changes obviously is because you're trying to measure the uptake of CO2 specifically. If you're measuring the uptake of CO2 specifically, you don't want temperature to be changing, because remember, temperature affects the rate of photosynthesis. If the temperature is high in the first 5 minutes and in the next 5 minutes the temperature is low, then your results cannot be said to be reliable. So you want to ensure that you're only measuring CO2, which is more or less your only variable. And then also you want to ensure that the temperature does not affect the rate of the reaction by switching it to a higher rate or to a lower rate of reaction. A third thing that you might also want to put in mind is that when temperature is high, you have what we call photorespiration. And remember, if you've studied C4 plants, you will remember that this is what happens with them when the temperature is high. Instead of them undergoing the Calvin cycle, they start to undergo photorespiration because high temperatures support that. So that is how you'd answer that question. So you want to ensure that you only track CO2, you want to ensure that the temperature does not affect the rate of the reaction, and you also want to ensure that photorespiration does not happen. Let's look at question 2. All organisms respire to release energy from energy-rich molecules, such as glucose. Anabolic reactions use energy. State 2 examples of anabolic reactions occurring in living organisms. Anabolic reactions mean, anabolic means that you are building up. So always bear that in mind. Catabolic, so catabolic is the opposite of anabolic, and what catabolic means is that you are breaking down. All right, so when you eat food, for example, so let's say you have a piece of beef and some vegetables, that food goes into your stomach and it is broken down into different constituents so that your body can absorb it. That is a catabolic reaction. An anabolic reaction is one whereby you are building things together. So examples of anabolic reactions might be to maybe build a protein within your body. It can be maybe DNA as well, building your DNA. It can be what we call phosphorylation, whereby you're adding phosphate to something. So again, anabolic reactions refer to adding things together to make one thing. Catabolic is sort of like breaking things apart. Okay, then let's look at the second part, A2. Describe and explain the use of ATP in glycolysis. Glycolysis is the first state, or the first stage rather, in respiration. So if you haven't watched the videos, please again go watch that. I don't want to go over it unnecessarily, but go watch the videos on respiration and you'll find glycolysis there. What ATP does in glycolysis is that it donates a phosphate group that is then added to glucose in order to make a glucose phosphate. Okay, it also then ensures the second what's it called, the second phosphorylation as well. So you have glucose or you have fructose. Here's what would happen. You have glucose is converted to fructose through an isomerase enzyme, and then that is phosphorylated. Okay, so it goes to fructose, and then you add a phosphate group. So I'm just going to write P over here. Then you have a second phosphorylation, which is where you then make fructose bisphosphate in glycolysis. So it allows for those two phosphorylations. Also, it ensures that by adding phosphates to glucose, it activates the glucose and prevents the glucose from being able to leave the cell. In this case, you only have to write three things, so any one of those three will do. Let's look at B. Some factory workers in the early 20th century were exposed to chemical X and experienced serious side effects. Some of the effects of exposure to chemical X are decreased production of ATP, increased lipid metabolism with weight loss, increased production of pyruvate and lactate, excess heat energy release causing an increase in body temperature which can be fatal. Chemical X increases the permeability of the inner mitochondrial membrane to protons, causing some protons to leak out into the matrix. Now again, this is an example of how CIE would ask you questions if you're writing the exams. So it's not enough for you to simply just memorize things with the hope of regurgitating them. You have to understand how those things apply or how they would apply in a new situation. So let's look at what question 1 says here. Explain why people exposed to chemical X show decreased production of ATP and increased lipid metabolism. So in this case, decreased production of ATP is due to this leak over here. If you remember, during your ATP synthesis stage of respiration, you have what we call a proton gradient, whereby protons are deliberately pumped out into the, not into the matrix, into the mitochondrial membrane, and then they are brought back in through a proton pump, and through that process of proton movement, you would have chemiosmosis, which then leads to the formation of ATP. In this case, the protons are leaking into the matrix, which means that there would be no steep gradient for the protons, which then means that you might not be able to have the ATP synthase happening or the chemiosmosis happening. In terms of increased lipid metabolism, what is happening in this case is that the body is now using lipids to produce energy because there is maybe less ATP that's being made from glucose. And the reason why you can say there's less ATP being made from glucose is that you see here it says increased production of pyruvate and lactate. Lactic fermentation is an indication that there isn't sufficient oxygen in the body to make ATP from glucose. Lactic fermentation can also be a pathway that is followed if your body is respiring through lipids. All right, so in that case you would be able to use that as a backup response. Then the second question says, suggest and explain why chemical X causes increased production of pyruvate and lactate, which is more or less what I've just mentioned. You have an increased rate of glycolysis. There is less oxygen in the body. If there's less oxygen in the body, then that means that your body would not go through respiration in its 4 different stages. There would be no link reaction, basically. Your body would simply stop at glycolysis, from which it would go through lactic from which it will go through lactic fermentation. So you'd have more anaerobic respiration, more or less. All right. And then this is the last one, suggest why excess heat energy is released. So in this case, because the body is making less ATP, which is what the question has already told you, it means that all the extra energy that's being made from the fermentation processes or even from whatever other process is happening is being released as heat because it's not being used to make ATP at all. So in this case, it would be released as heat. Let's look at question 3. And I know this is going a lot quicker than some of you might expect, but if you're writing A2, there's a certain level of responsibility that I ascribe to you as a student. I hope that you have done your homework and you have studied hard enough to be able to follow very quickly what is being taught, and to also refer to your notes if you get stuck. Obviously, you can always post a comment. If you're really stuck, you can post a comment and I'll try to respond to you. But I hope that you're able to catch on as we are going through this paper, and you can catch on with ease. Okay, in 2014, scientists published a phylogenetic, which is an evolutionary tree diagram, showing how and when different groups of insects developed from ancestral groups. To construct the diagram, the scientists compared nucleotide and amino acid sequence data for 1,478 genes from each of 103 different insect species. The 103 species represented all 32 orders of the class Insecta. This is a question from, I believe this is chapter 18. Many students tend to skip this chapter because they're always like, well, who's going to ask me about that? Please note that CIE will ask you the last 3 chapters in your A2 syllabus. So your A2 syllabus is from chapter 12 to chapter 19. The last 3 chapters, 17, 18, and 19, are just as important as the first 4 chapters, which are like 12, 13, 14, and 15, and 16, the first 5 chapters rather. Students find those chapters interesting because they're more relatable, and this ones tend to talk about things like evolution, speciation, and so on, and students just tend to tune out. Please don't tune out. By all means, make sure you study as much as you can, all that you need to know here. So yeah, the first question, A1, says outline 3 ways in which the members of a species are similar. So this is something that you can simply just get from the definition of species. So they can be similar in terms of like their genetics, they can be similar in terms of behavior. Similarity also is being grouped together as a species means that 2 organisms from that group can mate with each other and produce fertile offspring. So that's also something. So just keep that in mind. Then, so just again, the 3 ways, it says outline, so you don't need to write a whole list. Outline simply means you list, you don't need to write a whole prose rather. Outline means you just list out, so you can just write here, they can be similar morphologically, they can be similar genetically, they can breed with each other to produce viable offspring, and all of that. So that would give you your response. Organisms are classified in a hierarchical system of taxonomic units. Name the domain and kingdom to which insects belong. Insects are eukaryotes, okay? So in this case, the domain for eukaryotes is Eukarya. And I'm pretty sure many of my students who wrote the May June 2021 exams probably got here and thought, what? Why would they ask us this? Well, here we go. And the kingdom that insects belong to is Animalia. This is not something that I've deduced in any particular way. This is just a fact. You think of the organism that you're being asked about, and obviously if you know what the domain, kingdom, order, class, and all of those things are, then it makes your life a lot easier. The last question on the slide, it says, explain why a phylogenetic tree is useful for classifying insect species. So in this case, phylogenetic trees actually show the relationships between different species. So you can state here that it shows the relationship between closely related species. It can also help you to identify the different families within that. So even though you can have certain species, especially with things like insects, you would find that even though they belong to the same species rather, they still have like differences in terms of like order, class, and all of that. So phylogenetic trees are very helpful for that, and they also help you to see if certain species have the same ancestor. So that's one of the reasons why they would use a phylogenetic tree. Okay, let's look at question B. It says here, for this study, new software for processing large quantities of data was developed. Calculate the total number of sequences that were compared, considering both the nucleotide and amino acid sequence for each gene. Then it goes on, I put this here actually, so this here is a snapshot from the question earlier, just so that you can have something to refer to. It says to construct the diagram, the scientists compared nucleotide and amino acid sequence data. So that "and" is in bold because obviously they want you to take note of that, for 1,478 genes. So in this case, they're telling you that for each gene, there were 2 different sequences that were compared. So they compared the nucleotide sequence and the amino acid sequence. So that means you have 1,478 times 2. All right. And obviously for the different species, you had 103 different species. So that means you multiply that also by 103. And I think when you do that calculation, let me just check quickly on my calculator over here, you will get 304,468. So that would be the answer here in this case. So again, in case you are confused, let me just explain that really quickly. The question asks you, calculate the total number of sequences that were compared, considering both the amino acid and nucleotide sequence for each gene. Now, in the question earlier, you were told that to construct the diagram, the scientists compared the nucleotide and amino acid sequence data for 1,478 genes. That means they took 1,478 genes, compared their nucleotide sequence, then they took those same genes, compared their amino acid sequence. So they were compared twice. That is why you have 1,478 times 2. Okay? And then not only were they were compared twice, but they were compared for 103 different species. So not just one species. If it was just one species, then the calculation would stop with times 2. But because we have to calculate the different species as well, then that gives us this calculation, which then leads us to the value of 304,468. Okay. Explain why this study needs to make use of bioinformatic techniques. Bioinformatics, this is just asking you what bioinformatics is used for. And again, this is an unusual question. In the past, I think 3 to 4 years, this is not a question that has come up a lot. So if you're writing in October, November, please take note of these things. Bioinformatics is usually used to process large quantities of data. That's already been told to you in the previous question. It's also used to compare sequences. So if you have different species, for example, there is a tool called BLAST that geneticists would use that would enable them rather to tell the difference between different species by giving them more rich analysis of their data. Bioinformatics is also faster. Like you just saw now, we had to do all this calculation by hand. With bioinformatics, processing the data is a lot quicker and a lot more efficient. So those are the 3 things you would need to write there to get full marks. Let's look at question 4 very quickly. It says here, genetic engineering involves the manipulation of naturally occurring enzymes and processes. List the enzymes used in genetic engineering, that's genetic modification, and outline their roles in the natural process. So here you have 5 marks, which means that you need to list 5 different enzymes. So if you've watched the videos that I did on chapter 19, this is a chapter 19 question. This should make sense or should go very quickly. So you use what we call the restriction enzymes. I'm not going to write what they're used for, but the restriction enzymes are used to cut DNA. All right, that's their role, and they cut DNA at specific points or specific triplet codes, depending on what it is that you're dealing with. So they used to cut DNA. You also have what we call what's it called now, our reverse transcriptase, if you're dealing with RNA. So your reverse transcriptase will ensure that your RNA is transcribed back to DNA. Also, when you cut your DNA during just trying to write transcriptase here quickly. When you cut your DNA during genetic engineering, it's important for you to bear in mind that you don't just leave it cut and open. So you don't cut a DNA loop, for example, and make it a linear thing and expect to keep working with that. So ligase is an enzyme that's very important, and ligase helps you to join cut DNA back together. Okay, so ligase is used to add DNA back together or join the different fragments back together. You also have what we call the Taq polymerase, and again, this is not asking you specifically for one type of genetic modification. If you remember, when I did the videos on genetic engineering, we spoke about PCR, we spoke about gel electrophoresis, we spoke about microarrays as well. So in this case, you are simply listing the different enzymes that you would use in all of those things. All right. So here you have Taq polymerase. Taq polymerase is used in PCR, and it is an important one for the elongation step of the PCR. All right, so that's what you would use that for. Reverse transcriptase is used in microarrays. Restriction enzymes are used when you're doing specific types of genetic engineering. I'm just trying to think of one more enzyme that you might need, but I'm not really sure. But in this case, since it says list the enzymes and outline their roles, you might get 2 marks for a specific role, and maybe that would be helpful in that case. Let's look at question B. Genetically modified organisms, so that GMOs, include crop plants. Genetically modified crop plants have been grown in North America in increasing quantities since 1996. They are now grown in many areas of the world and are eaten by millions of people and farm animals. Table 4.1 compares the area of land used to grow GM crop plants on 6 continents in 2013. So you can see here in Africa it is 3.5 million hectares, and it's usually maize and soybean. In Asia, it's 19.1. In Australasia, so that's the part of the continent, Australia and Asia, the Asian extension of that is 0.6. Europe 0.2. North America 81.1, South America 70.9. So obviously from this data, if we were to go by this data, if you don't want to eat GMOs in every single thing, it seems like Europe is the place for you to go and live. But that is a digression. Let's look at the question. Suggest how North America and South America benefit from growing large areas of GM crop plants rather than non-GM crop plants. Well, this is just asking you for the advantages of genetic engineering in agriculture, which is one of the videos I recently uploaded. Obviously, it leads to an increased yield. Also leads to reduction in the use of pesticides for certain crops, but not necessarily. Sometimes it also leads to like decreased decreased risk of failure, of crop failure. So if you have increased yield, it obviously then translates to the fact that the farmers will always have more for them to sell, and that means that their profits are secure. And also, so I'm just going to write it here. Actually, I should do what my students do when they're too lazy to write, and that is they draw an arrow. They would do something like this, and they would say increase. So I'm supposed to translate that as increased yield. I usually diss them for that, but look at me using it now. So increased yield, obviously, increased profit for the farmers, okay, or security, or increased security for their crops, decrease in what's it called now, in pest attacks or pesticide use, pest attacks or pesticide use as well, because some of these GMOs are able to produce their own pesticides. So for that reason, you don't need to use pesticides. So actually, I would just say decreased use of pesticide, and that would give you 3 marks there. Okay, and I think I've seen this question before, by the way. I think this was written 2 or 3 years ago as well. All right, only a small area of land is used to grow GM crop plants in Europe. This is mainly because most of Europe uses a precautionary principle. The precautionary principle prevents GM crop plants being grown if there is a possible risk of harm to human health or to the environment, even if there is no proof of harm. Many European citizens have concerns about the safety of eating GM food, but Europe imports large quantities of GM maize and GM soybean to feed farm animals. Use the information and table 4.1 to deduce 2 arguments in favor of growing more GM crop plants in Europe. Well, in this case, something that you might say, okay, well, as proof is that you can look at the number of people that, or the amount of land that's being used to grow GM crops in other areas. And so you can then say, well, in this case, let's think about it. If this amount of land is being used and this amount of crops are being grown, then obviously more people are eating GM foods, and so they seem to be healthy. Well, that would be your argument. But again, this is a very touchy issue. For me, this is not a right or wrong question, to be honest, but it seems as though CIE wants to make it right or wrong for this particular paper. So I guess if you're deducing an argument in favor, you'd speak about the fact that in other continents, quite a number of people consume GM crops. And that should be an indication of its safety. Already Europe is importing GM crops to feed its farm animals, so it would only make sense for it to grow those crops and maybe save costs on importation. So those would be two arguments that you can put forward for this one. Oh, there's one more question right here in the bottom corner. I don't know why I fit everything in here, but well, let's just go for it. Explain why the data in table 4.1 are not enough to calculate the extent to which different continents have replaced traditional crop plants with GM versions. Well, I mean, that's pretty straightforward because it simply tells you that the area of land used to grow crop plants are this much, but you don't know what the full area of land for those continents are. So you can't really say that they're being rejected. So just because they use 81 million hectares of land in North America doesn't mean that nobody is growing traditional crops anymore, because for all you know, the land size might be maybe 3 million hectares, not 3 million, 300 million hectares. So you're not really sure. It could even be 3 billion hectares. You don't know that for sure. So you can't work out the percentage of land that's being used for that reason, so you would not be able to say it's not enough for you to calculate anything. Okay, so for the sake of not making the papers the videos unnecessarily long, because I feel like when the videos are like up to an hour, most of you don't follow through to the end. I'm going to stop this one here, but please watch part 2 for the rest of the paper. So please don't miss out on that. Make sure you watch part 2. All right. And also something I should have said at the beginning, it's always better if you've done these papers ahead of time before getting into the videos to see the explanations. So just make sure you do that.